Hello students, welcome to EPG Pathsala. I am Professor Praveer Kumar from Delhi Technological University. Today we are going to discuss about a module of polymerase chain reaction in short PCR under the paper Genetic Engineering and DNA Recombinant Technology. So my talk will be divided into several parts. First is introduction to PCR. What are the different steps and procedure? Then primer designing. What are the key components and the limiting factor? And finally the summary. The polymerase chain reaction is a very important and powerful technique in molecular biology where you can amplify even the tiniest molecule of tiniest concentration of DNA into an appropriate amount or in concentration. Here, the DNA amplification is in exponential growth, and after attaining a certain cycle, you can have the appropriate concentration of DNA. This is an automated process, and with the help of thermocycler, we are amplifying the DNA under different conditions, that is the denaturation condition, annealing condition and elongation. The procedure of PCR is typically consists of several cycles, such as from 20 cycles to 40 cycles with the repetitive change in the temperature, and this repetitive change is called cycle. Each cycle is commonly consisting of 2 to 3 different discrete temperature steps, and finally we are doing the PCR with the 30 cycles or 40 cycles. In the PCR, the first step is denaturation, and this is also called the first cycle, while the last cycle is the elongation, which I will explain these things in the later part of my talk. So the denaturation temperature usually 94 to 95 degrees centigrade, while the elongation is at 72 degrees centigrade. So I will explain all these things in the later part of my talk. PCR, which is polymerase chain reaction, is an in vitro process by which a DNA is used as a template, and along with DNA we need the internal primer or primer per se from the both direction, that is from the forward direction as well as in the reverse direction, together with PCR buffer, DNTPs, magnesium chloride and Taq polymerase with the PCR grade water. PCR first described in mid 1980s by Mullis, who was also the recipient of Nobel Prize in 1993, where a PCR is amplifying with the help of Taq polymerase or a polymerase which can sustain at high temperature, which is derived from the bacteria Thermophilus acuitas. An in vitro method for enzymatic synthesis of a specific DNA sequence, and which amplify the DNA from the heterogeneous pool of complex DNA or cDNA probes. What it requires, as I discussed, it requires primers, Taq polymerase, buffer, DNTPs, template DNA, and of course a thermocycler. So let's discuss the PCR cycle in brief. First step is denaturation. What does it mean? The target sequence, which is first denatured at higher temperature, so the double-stranded DNA is converted into the single-stranded, or you can say that double-stranded of DNA is separating and becoming the single-stranded DNA. The step 2 is the annealing, where the temperature is low, and primer, which has a suitable temperature or an appropriate temperature of primer that has been calculated based on the TM temperature we can use. And this second step is primer annealing, where primers are bind at the 3' end of the template DNA and anneal. The third cycle is elongation. With the help of Taq polymerase, the primer start sliding on the template DNA with the help of DNTPs at 72 degrees centigrade, and finally at the end of first round of PCR cycle we can get 2 copies of target sequence. So if you can calculate, the number 1 cycle is giving you 2 copies, second cycle is 4, third cycle is 8, fourth cycle is 16, fifth cycle is 32, 6 is 64, and after that 20 and 30th cycle it is in the million number of amplicons of the target. As you can also see in the slide, the PCR is having the directional synthesis, that is 3' to 5' in both the orientation. So here is a typical thermocycler, and you can also call it as Xeroxing or Xerox machine of a DNA. And as I explained in the previous cycle, that what is happening in the 2 copies become 4 copies to 8 copies and so on and so forth, and at the end you can have millions of copies, and that within a very short time span. So if we can conclude these portions, we can say PCR is divided into 3 stages, where first stage is denaturation of double-stranded DNA into single strand, and the timing of denaturation is very, very important. If DNA template is originated from plasmid, then 3 to 5 minutes of denaturation is adequate, whereas if DNA template is of genomic in origin, that is the genomic DNA, the 7 to 10 minutes of first denaturation is always recommended. Most importantly, here I would like to explain that in the plasmid DNA, the DNA template concentration should be 10 to 20 picogram of concentration, but in case of genomic DNA, we must take at least 0.5 to 1 microgram of DNA as a starter material. And this is the called first cycle of the denaturation. In avoid to renature the 2 single-stranded DNA, a short denaturation period of 30 seconds to 2 minutes is advisable from second cycle to the penultimate cycle, where a short denaturation is followed by annealing and elongation. What is happening in the final stage? At the final stage, the last elongation is required in order to complete any product which is not elongated properly. Once the final cycle is completed, the entire reaction rests at 4 degrees Celsius till the use of the product. Now, if I can say that this PCR cycle is 30 cycles, what does it mean? The first cycle will be for the complete denaturation. Cycle number 2nd to the 29th cycle, it will be the denaturation followed by annealing followed by elongation, while the last cycle, that is the 30th cycle, is final elongation. Here is a depiction of simple thermocycling protocol that you can see that denaturation, again denaturation, annealing, extension, and finally it holds. Now come to the PCR curve. As I have shown you here that this is a typical amplicon production and an exponential phase, a log linear phase, and then a plateau phase you can achieve at the end of the cycle, where there will be no more production of amplicon synthesis. So I have already explained here. You can also see denaturation, that is step 1, annealing, that is step 2, and extension, that is step 3. Now come to the components which is necessary for the completion of a polymerase chain reaction cycle. And these are the template, oligonucleotide primers, DNTPs, thermostable DNA polymerase, magnesium chloride, and buffer. And in the buffer, the working concentration of buffer is having potassium chloride, TrCl, and sodium chloride. Before doing any PCR reaction, what we are doing, we are preparing a master mix. And the master mix where, apart from DNA template, we are mixing all the components in one tube and equally distributing in different tubes wherever there is DNA. So the master mix usually consists of forward primers, which is 10 to 20 picomole in concentration, reverse primer, again 10 to 20 picomole concentration, DNTPs 10 to 20 nanomole concentration, 1x PCR buffer, 3 units of Taq polymerase, and to make the volume appropriate, PCR grade water is required. So if we can say what are the various limiting factors in PCR, is first and foremost the DNA template concentration, length of the PCR, then concentration of primers as well as the DNTPs and Taq polymerase. Now come to the PCR primer. PCR primer is very important component to perform PCR cycle. And here there are again the limiting factors such as primer length. Normally the primer length should be 20 to 30 mers. Mers means nucleotide, which has optimum annealing temperature, and it is also necessary that the annealing temperature should not be exceed more than 65 degrees centigrade. However, one can use the desired length of primer, but at the same time one should also look what is the efficacy of that primer in terms of annealing temperature, and second, it should not cross with the elongation temperature. Another point is the primer length is the length between the forward primer and the reverse primer should not be much varied, which means if a forward primer is 15 mer and the reverse primer is 30 mer, this may cause a problem during the amplification procedure. And that is why it is always recommended that the both length of primer in the forward direction as well as in the reverse direction should be more or less equal number of nucleotides. Now come to the annealing temperature. In annealing temperature, the forward and reverse primer is always binding to their 3' complementary sequence, and therefore annealing temperature is very crucial. First of all, the annealing temperature is determined by 4GC plus 280 formula, through which we can decide that what is the approximate annealing temperature of a primer. Now come to the second point, primer-dimer formation. Sometimes a long primer has repetitive sequences, and therefore while the amplification process, these primers may form a loop or hairpin, and sometimes primer-dimer formed. So we should avoid the primer-dimer formation. Next, come to the GC content. We must calculate melting temperature of both the primers, that is the forward and reverse primers, and in case of the primer length, which is approximately 20 to 30 mer, make sure that in the both the flanking region, that is at the 5' end and the 3' end, there has to be G-series sequence so that it can easily anneal due to the 3 hydrogen bonds. Further, make sure while designing a primer that flanking region of primer should have sequence which is GC rich. It will also provide stability while annealing. Sometimes if GC content is too high, the rate of reaction or kinetics may be slow due to the adherence of G-series sequence to its complementary sequence.
So now I would explain what are the different types of PCR. I have already explained to you about the hot start PCR, the nested specific PCR, intergenic PCR, multiplex PCR, colony PCR, reverse transcription PCR. So hot start I have already explained that after first cycle of denaturation, we are adding the Taq polymerase. What is happening in the nested specific PCR? Nested primers are gene specific primers, which means if gene sequence is unique and only present in one species, it can come under gene specific application or nested PCR. In this PCR, a specificity of DNA amplification is increased by attenuating non-specific amplification of template DNA. In this PCR, two primer sets are used for two successive PCRs, where the first round of PCR, first set of primer, is generating amplicon, which comprises of non-specific amplicon, and this amplicon is act as a DNA template for the second round of PCR with the second set of primer, and which is gene specific primer. That means the primer has 100% sequence similarity with the complementary DNA sequence. Come to the intergenic PCR. If the sequence similarity is very high among many species, we have to design primer in such a way that minimum mismatches area is to be identified. To achieve this, multiple nucleotide alignment is performed among multiple species and identifies the conserved region at 5' and 3' ends, and in one go we can amplify many genes with the same set of primers. Come to the multiplex PCR. This is a combinatorial approach where multiple set of primers can amplify different amplicon, but the gene fragment or the DNA template is common. Usually, if we want to amplify 3 to 5 genes at once, forward and reverse primer of all 3 genes are designed in such a way that neither it crosses the annealing temperature or cross interference with the 2 primers. So the important thing here that the primer length, second, there is no overlapping sequence, and third, the same DNA template. The another important aspect of the multiplex PCR is that amplicon size from each reaction has to be different and has to be in a different physical length. Another limitation of the multiplex PCR is the concentration of MgCl2. What is happening? Normally, the 10x PCR which has been supplied by the manufacturers has already 1.5 millimolar of magnesium chloride. However, in case of the poor amplicon production, we may increase the concentration of MgCl2 up to 3.5 millimolar, and thus it is recommended that the amount of magnesium chloride in the multiplex PCR should not exceed from the desirable amount, otherwise it may produce many non-specific binding and thus the product. Now come to the colony PCR. This is also a quick technique through which from the bacterial plate you can lift a colony, and that colony will act as a DNA template with an appropriate amount of primers, DNTPs and Taq polymerase, and in one go you can identify whether that colony is the product of recombinant or not. Now come to the reverse transcripted PCR, that is the RT-PCR. This PCR is also used for the expression profiling, where reverse transcription is used to prepare cDNA from the RNA. With the help of reverse transcriptase enzyme and random hexamer primers, RNA is reverse transcribed into the cDNA, and then it can be used for the various purposes.
So now come to the thumb rules of the PCR conditions. And as I said you that the first cycle, which is the denaturation cycle, sometime if the single strand separation is not an appropriate or desired, we can add some extra minutes in case of longer template to the cycle and ensure that the entire template is denatured. There is a phenomenon which is recently came in picture, is the hot start PCR. Initially, scientists was having this notion that during the first cycle, which is entirely attributed to denaturation, the Taq polymerase was working nothing at that. Taq polymerase is working nothing in that particular cycle. So scientists were thinking that during this 5 minutes or 4 minutes of denaturation, or sometime in 10 minutes of denaturation, in case of genomic DNA, that Taq polymerase is doing nothing but only sitting there. So they have thought that that might reduce the efficacy of Taq polymerase, and that is why they have started a concept of hot start PCR. What is happening in the hot start PCR, that upon completion of first denaturation cycle, that is after 4 to 5 minutes in case of plasmid DNA, or 7 to 10 minutes in case of genomic DNA, you stop the reaction for a minimum period of time and add the Taq polymerase, because after first reaction, that is the first cycle, second cycle till penultimate cycle, a brief denaturation followed by annealing and elongation, and there Taq polymerase will immediately start working. So this was the concept of hot start PCR, that upon completion of first reaction, add the Taq polymerase and complete the reaction. Next, appropriate melting temperature. As I have already explained you, that melting temperature is very important. In case of the primer which has not GC rich sequence at the flanking ends, what would happen? The adherence capacity or the annealing capacity will be poor because of the non-GC nucleotide. And this is the reason why at every time, if you are constructing or designing a primer, you must keep GC rich sequence from both the flankings in the forward primer as well as in the reverse primer. Now, the calculation of annealing temperature is also important. Initially, 4GC plus 280 content will give you a rough estimation of annealing temperature, but it cannot be an appropriate temperature, and in order to avoid this, you must set up a gradient PCR. Now, what is gradient PCR? Suppose if you are confused between the annealing temperature of two different primers, that is the forward primer and reverse primer, what you can do, you just set the temperature and annealing temperature on gradient PCR starting from, let's say, if one forward primer melting temperature is 52 degrees centigrade, and if the reverse primer is 45, what you can do, you can make a gradient from 50 degree, 52 degree, 54 degree, 56 degree, 58 degree and 60 degree temperature for annealing, and do the gradient PCR. By doing this, what you can obtain a suitable range of melting temperature, that is the annealing temperature, where you will have the best PCR product, and then you can use that temperature for your future experiments with the same set of primer. Now, what are the common PCR additives? BSA. Whenever we are doing the PCR, BSA is already in the buffer, which is usually into 0.1 to 0.1 microgram per microliter concentration. What is the function of BSA? BSA is stabilizing the Taq polymerase and overcome the PCR inhibitors. Now come to the DMSO. DMSO is usually 2 to 5% volume by volume, and what it doing? It is doing the denaturation. A good keeping of GC rich temperature, primer strand from forming secondary structure. So DMSO can prevent this. Now come to the glycerol. This increases the apparent concentration of a primer or template mix and often increases the PCR efficacy at high temperature. Now come to the formamide, beta-amide or TMAC. The concentration used vary by different type of PCR Taq polymerase, and there are also some non-ionic detergents such as Tritonax, Twin 20 or nonidate P40 in 0.1 to 1%. So these are the different additives which you can use in a common PCR. The most important is magnesium chloride, because suppose if a DNA has low copy number and in the end you are getting a very faint product or faint amplicon, you can enhance this efficacy, I mean the amplicon product, by adding some magnesium chloride. Magnesium chloride is enabled to give the better binding, and you can have in the end the better product. But apart from the better product, you will have many other non-specific bindings as well.
Now come to the different problems in the PCR which you might encounter while performing an experiment. It is very important to use 2 to 3 negative controls all the time to validate the efficacy of a PCR reaction. However, there are many more factors which is important for the unusual amplification or altered amplicon generation, such as the template concentration. If template concentration is in appropriate range, there will be an erroneous amplification and the resultant amplicon is really not properly visible. Annealing temperature. If annealing temperature is varied from reaction to reaction, or if the primer is too big or too small, it can also interfere in the PCR reaction. Extension time. This is another important point, and suppose if it's too short, increase an extension time for the longer product. Cycle number. Cycle number is also very important. In order to obtain an appropriate concentration of amplicon, cycle number has to be determined before starting the PCR reaction. The primer designing, as I already explained you, with the help of bioinformatic tools, or if not, you can also do it manual, but make sure the primer length, GC-rich content, annealing temperature, inhibition of the primer dimer, these things you should keep in your mind. Come to the last point, that is non-specific priming. If the primer set is not binding properly with given DNA, sometime hot start PCR is advisable. So these are the different troubleshoot points during the PCR. Now come, if template concentration is inappropriate, just optimize by titration. If extension time is inappropriate, cycle number is too high maybe, and then you have to reduce the cycle number. Primer designing is not appropriate, then go with the concentration. Non-specific priming, again optimize by the titration. MgCl2 concentration is too high, again optimize by the titration. If the GC-rich template, then use the PCR additives. As I have already explained in my previous slide, that suppose if a sequence is GC-rich, you should add the additives. Next one, problem with the thermocycler. To troubleshoot it, go for the positive control reaction. If the enzyme concentration is low, optimize it by the concentration. Annealing temperature is low, again optimize it by the gradient PCR. Cycle number is low, then again you should manipulate the cycle number. Primer concentration is high, go with the titration experiment. So before doing any PCR experiment, you must go for a pilot PCR, and in which you can at least get a clear view that what kind of modification require in a thermocycler.
To summarize, PCR is a very powerful technique in the molecular biology where you can amplify even a tiniest amount of DNA with the help of suitable primer. The application of PCR is many places, like in forensic medicine, in DNA fingerprinting. You can also have the PCR for the cloning. Suppose if you want to make any changes in the cloning, you can also use the restriction sites on primer, and you can use it while the cloning procedure. So it can be very effective tool in molecular biology, and that is why PCR is an important tool. Thanks for your attention.